The size of most cells and organelles makes it impossible to see them with the naked eye. Cells and their organelles are measured in micrometers, and as I mentioned previously, a micrometer is equal to a millionth of a meter. Structures that are in the micrometer range can generally be visualized using a compound light microscope. Molecules like the DNA double helix and subcellular structures like ribosomes in the cell membrane and many viruses are in the range of nanometers, and a nanometer is a billionth of a meter. Now, these structures cannot be visualized using standard light microscopes. However, there are certain microscopes, which we'll discuss later, that allow us to visualize some of these molecules and subcellular structures. The compound light microscope was very important early on in cell biology because it allows scientists to visualize cells and organelles, and it continues to be important to this day. This type of microscope consists of a series of lenses that enlarge the size of the specimen and a source of visible light. For a typical light microscope, the limit of resolution is 200 nanometers, and that allows for magnification of 1,000 to 1,400 times the size of the object that is being visualized. Now, the limit of resolution refers to how far apart two objects need to be in order to be seen as two separate points rather than as a single point. So a limit of resolution of 200 nanometers means that there must be a distance of at least 200 nanometers between the two objects to be seen as two separate points. If the objects are closer to each other than 200 nanometers, you would not be able to see them as separate points using a microscope that had a limit of resolution of 200 nanometers. So the lower the limit of resolution of a microscope, the better the microscope is, since it will have a higher resolving power, which is the ability to see fine details. And the limit of resolution of microscopes is determined in part by the wavelength of the light that is used for illuminating the sample. So shorter wavelengths are going to provide lower limits of resolution or better resolving power. In order to visualize a sample, it has to be placed between the light source and the lenses. So that means that in order for the light to pass through the sample has to be very thin. And for that reason, pieces of tissues have to be cut into thin sections to be visualized. In addition to being very thin, samples cannot be opaque, as that would prevent the light from passing through the sample. But that can be a problem if a sample is completely transparent, because we won't be able to see much detail. So there are ways of dealing with that. So let's look at the different types of light microscopes and discuss what they're useful for. The type of light microscope that most of us are familiar with is the bright field microscope, which is the one that has a white light as a source of illumination. Now, because a white light is used to illuminate the sample, this type of microscope is best for samples that are stained with dyes. However, dyes or the procedures that are used to dye tissue tend to be toxic. So that means that in general, bright field microscopes are not useful for visualizing live specimens. If we need to visualize live cells, there are certain types of specialized light microscopes that can allow us to do that. The phase contrast and the differential interference microscopes take advantage of the fact that when light passes through a specimen, different parts of the specimen will refract or bend the light in different ways. Now, the phase contrast and differential interference microscopes exaggerate these refractions so that different parts of the cell look different. And that allows us to see a little more detail, as you can see in these images, without having to actually stain the cells. So these types of microscopes are useful for visualizing living cells. However, since this is a type of light microscope and it has a limit of resolution of 200 nanometers in general, it would not allow us to see fine details. So structures like ribosomes, which are about 20 nanometers, could not be visualized using this type of microscope. Another type of specialized light microscope is the fluorescent microscope, which can be used to visualize both live or preserved specimens, depending on how the sample is prepared. Fluorescence microscopy is most useful for detecting specific molecules in cells, like specific proteins, which can be made to fluoresce in different ways. Now, I want to emphasize that this doesn't mean that we can see the structure of the protein, just detect its location. So as you can see in these images, we can label different structures in cells by using fluorescent dyes or some sort of antibody staining or genetic labeling with green fluorescent protein. That doesn't mean we're going to see the structure of the protein, but we will be able to see whether it's present in the cell or not, and we'll be able to label specific things, organelles, like the nucleus, for example, by labeling a protein that's present within those organelles. Now, one way to make a molecule fluoresce is to use a fluorescent dye that is designed to bind to specific proteins in cells. For example, the toxin phalloidin, which is produced by some mushrooms, binds to actin. If phalloidin is chemically conjugated to a dye, as shown here, it can be used to label actin filaments like the ones shown in these images. It is also possible to genetically incorporate the fluorescence, so you can use green fluorescent protein or GFP, but you have to use a genetic technique in order to make sure that wherever your protein of interest is, your fluorescent protein is also made, so that you can localize the location of the protein in cells or tissues. Now, incorporating fluorescence genetically will allow you to visualize living cells. Finally, cells can also be made to fluoresce using antibody staining. Antibody staining takes advantage of the fact that vertebrates develop antibodies in response to the presence of a foreign molecule, which is known as an antigen. Antibodies are proteins, and they contain a constant region, which is the same for all antibodies of a particular type, and variable regions, which are the regions that recognize and bind to the antigen. Now, an antigen could be any molecule, but it could also be a protein. And that means that we can make antibodies against protein of interest by injecting an animal like a mouse or a rabbit with the protein. Of course, the animal will only develop antibodies against foreign proteins. The antibody can then be purified, and it can be conjugated or covalently attached to a fluorescent dye. There are two types of fluorescent antibody staining: primary, which is also known as direct immunofluorescence, and secondary or indirect immunofluorescence. In primary immunofluorescence, only one antibody is used. Now, this antibody both binds to the antigen and also is conjugated to a fluorescent dye or fluorophore. On the other hand, in secondary or indirect immunofluorescence, two antibodies are used instead of one. The first antibody, which is unfortunately known as the primary antibody, is an antibody that binds to the antigen, as you can see here, but it does not have a fluorescent label. So we can't see anything if we just use the primary antibody. So this means that in order to visualize where the primary antibody has bound, a second antibody, which is known as the secondary antibody, has to be used. And this antibody has a fluorescent label. Now, the antibody, the secondary antibody, attaches to the primary antibody because the primary antibody is the antigen to the secondary antibody. Now, indirect immunofluorescence is preferred over direct immunofluorescence. So secondary immunofluorescence is preferred over primary immunofluorescence because it produces a brighter image. And the reason for that is that more than one antibody, as you can see here, can attach to an antigen. And since multiple primary antibodies can attach to the protein of interest, and then multiple secondary antibodies can attach to each primary antibody, the fluorescence signal is amplified. Although fluorescence staining allows us to visualize the location of specific proteins and to label organelles, there is detail that cannot be visualized with fluorescent microscopes. Now, there are super resolution fluorescent microscopes that allow us to visualize structures ranging from 50 to 100 nanometers. However, some structures like ribosomes, as you can see here, are smaller than this. So electron microscopes are needed for these types of structures. And the electron microscope uses a beam of electrons as a source of illumination rather than light. Now, since the wavelength of the electron is much shorter than the wavelengths of both visible and ultraviolet light, electron microscopes have a limit of resolution that is 100 times better than light microscopes. So their limit of resolution is about 2 nanometers. That means that they can magnify an object up to 100,000 times its size. Now, there are two major types of electron microscopes. In transmission electron microscopy, the beam of electrons is passed through the specimen, and this type of microscope is best for visualizing intracellular details. For example, the cristae in a mitochondria, which are enfoldings of the inner membrane of a mitochondria, can be visualized using transmission electron microscopy. In scanning electron microscopy, on the other hand, the electrons bounce off the surface of the specimen and the image that is produced is three-dimensional. Now, many electron microscopes require samples to be dehydrated, so in general, they're not suitable for live cells. However, there are newer electron microscopes that allow for cells to be visualized in a liquid medium. Another technique that is essential for cell biologists is cell culture because it allows the study of cellular processes in more controlled environments. Although many cells can be isolated and grown in culture, the majority of human cells cannot grow indefinitely in culture. So most cells divide about 50 times before undergoing apoptosis. The reason for this is that every time a cell divides, its telomeres get shorter. Now, the telomeres are the sequences at the ends of chromosomes that do not code for protein. So, because these telomeres serve to protect the protein coding region of chromosomes, the loss of the telomere sequence, which happens over time as cells divide, triggers apoptosis. However, cancer cells express an enzyme known as telomerase that extends the telomere sequence, and that allows cancer cells to divide indefinitely. The fact that cancer cells can divide indefinitely was discovered in the 1950s with the isolation of HeLa cells, which came from a cervical tumor of a cancer patient, Henrietta Lacks. HeLa cells are widely used for research because they not only divide indefinitely, but they also divide very rapidly. Now, it should be noted that Henrietta's family was not asked for permission to use these cells, and they were not financially compensated. In addition to that, the genome of HeLa cells was published without the permission from Mrs. Lack's family, but nowadays the family has gained some control of how these cells are used and how the information about these cells is shared. Now, since the isolation of HeLa cells, other immortalized cell lines have been developed, and these have either been isolated from tumors or they've been manipulated by either introducing telomerase or cancer-causing viruses. In the case of mouse cell lines, sometimes these cells can become immortalized spontaneously, but the reason for that is not yet known. Another technique that is essential for the study of cell biology is recombinant DNA technology. A recombinant DNA molecule is simply a DNA molecule that contains DNA from two different sources. In order to develop these recombinant molecules, restriction enzymes are used to cut the DNA at specific sequences. Now, these recombinant DNA molecules can then be introduced into a bacterial host using a technique that is known as transformation. And this allows the bacteria basically to serve like factories that replicate the recombinant DNA molecule. And that process actually is known as molecular cloning, which is different from the cloning of organisms. These recombinant DNA molecules can then be isolated, the transgenes can be purified, and they can be introduced into eukaryotic cells using a number of different techniques like transduction or transfection, or by simply injection of the DNA molecule into a zygote. And many transgenes contain reporter genes like LAC-C or GFP that allow us to visualize specific proteins because those reporter genes are placed under the control of the regulatory sequence for the gene of interest. In addition to being useful for studying when and where a particular gene is expressed, transgenic mice are also useful for studying the effect of overexpressing specific genes. For example, in this image, we see a mouse that expresses the rat growth hormone. Now, because the mouse already expresses its own growth hormone, the addition of the rat growth hormone means that this animal has higher than usual levels of growth hormone. And as you can see, the animal is larger than its normal or wild-type littermate, which does not have the transgene. Transgenic mice can also serve as models of human diseases. For example, certain cancers are caused by the overexpression of some genes. So we can develop a transgenic mouse that mimics the same condition that we see in the cancer. Another type of animal model that is useful in cell biology is the knockout mouse. Now, these mice allow us to determine the effect of the loss of a particular gene. Now, because certain diseases, including some cancers, can be caused by deletions of certain genes or by mutations which inactivate proteins, knockout mice can also serve as models of human disease. Now, keep in mind that a model is meant to closely resemble the condition that we're trying to study. Just like an architectural model is supposed to be a replica of an actual building. Knockout animals can also be used to figure out the function of specific genes based on the phenotype that we see. Now, for example, these pictures show a knockout mouse for the gene ETS1. Based on the phenotype of the mouse, what can you say about the potential function of ETS1 by looking at the phenotype of the knockout compared to the wild type? In addition to mice, there are other organisms that are also useful as models in cell biology. The most widely used include the bacterium E. coli, the arthropod Drosophila melanogaster, the yeast Saccharomyces cerevisiae, the roundworm C. elegans, and although not shown here, the plant Arabidopsis thaliana. Throughout the semester, we will encounter examples of how these organisms have contributed to the little bit we currently know, and will continue to contribute to the many things that we have yet to figure out.